We are performing the Hershey and Chase blender experiment, which was performed by Alfred Hershey and Martha Chase in 1952.
Hi. For the experiment, we have 2 different E. coli cells right here. So we are going to infect them with different bacteriophages. So in this E. coli cell, we are infecting it with bacteriophages containing phosphorus, which is represented by the green toothpicks.
And in this cell, we're infecting it with the bacteriophage that contains sulfur, which is represented by the red toothpicks.
The reason we are infecting it with phosphorus and sulfur is because phosphorus is only found in DNA, while sulfur is only found in proteins.
Yes. Now the E. coli cells are getting infected by the bacteriophages.
This is what the E. coli cells look like once they're infected with the bacteriophages. Now that these 2 bacteria cells have been infected with bacteriophages, the radioactive material is found in different places. For this E. coli cell containing the bacteriophages with phosphorus, the radioactive material is on the inside of the cell.
While in this E. coli cell with bacteriophages containing sulfur, the radioactive material is found outside of the cell on the protein coat.
This is the genetic material found inside the E. coli cells. This one is phosphorus, this one is sulfur. So now comes the part of the experiment involving a blender. So we're going to take this E. coli cell, which has the phosphorus bacteriophages in it, and place it in the blender so that the bacteriophages will be agitated enough to come off the outside of the cell. Successfully remove these bacteriophages from this E. coli cell.
Now I'm going to do the same thing to this cell containing sulfur that was previously done to the cell containing phosphorus. You can see that all the bacteriophages are successfully removed from the cell. Now we're on to our last step, which is centrifugation.
This is the centrifuge, and here is the E. coli cell containing phosphorus. We then place it into the centrifuge and mix it. The whole point of centrifugation is to cause heavy material like bacteria to move to the bottom of the tube and form a lump called a pellet. The lighter material flows to the top and creates a liquid layer called supernatant, which is not bacteria.
Okay, so after the bacteria is containing phosphorus is centrifuged, it will sink to the bottom since it's in the bacteria and it's heavier, while the lighter stuff, which is not bacteria, rises to the top.
Yeah. As we previously did with the E. coli cell containing phosphorus, we are now taking this E. coli cell containing sulfur and placing it into the centrifuge.
After centrifuging the cell containing sulfur, we found that it was lighter and rose up to the top because it's not part of the bacteria cell.
This proves that DNA is the genetic material. Recap of the Hershey and Chase experiment. In conclusion, the Hershey and Chase experiment is divided into 2 batches, each with 3 steps. The steps are the same for both in that the first one is infection, the second one is agitation or blender, and the third one is centrifugation.
In batch 1, step 1, the E. coli gets infected with the bacteriophage containing an isotope of sulfur. And the radioactive material stays on the outside of the cell on the protein coat. In batch 2, step 1, the E. coli cell gets infected with a bacteriophage containing an isotope of phosphorus. And the radioactive material is inside of the bacteria.
In batch 1, step 2, the E. coli cell is agitated in the blender to get rid of all the bacteriophages that are on the outside of the E. coli cell. This means that the radioactive material remains on the outside of the E. coli cell. In batch 2, step 2, the E. coli cell is agitated in the blender as well to get rid of all the bacteriophages on the outside of the cell, but this time the radioactive material goes into the cell.
In batch 1, step 3, after centrifuging the bacteria containing sulfur, you find the sulfur in the supernant, and the phages found inside the E. coli cells were not radioactive, which means that sulfur is nowhere to be found.
In batch 2, step 3, after centrifuging the bacteria cell with the bacteriophage containing phosphorus, phosphorus is still present in the cell. Once again, this proves that DNA is the hereditary material. Thanks for watching, and now for the bloopers!
After such...
Wait, you have to go. I'm not joking. As we previously...
So after the I said oh yeah, it is a bacteria. What are these?
Bacteriophage containing...
Now the E. coli cells are getting infected by the bacteriophages.
This is what the bacteria... No! Dang it, that was... Wait.